Okay, so here we go. There we are. So what I'm going to do today is just to talk you through at a relatively high level what the opportunities as far as translational medicine or translational science are in drug development, but also a reminder of the fact that there are certain things that we can't do. So the next slide. There we go. So what is translational science? And as I've already alluded to, it's also sometimes referred to as translational medicine. And essentially, it's applying applied research, preclinical, but also clinical research to support drug development. And this isn't a one direction from the preclinic into the clinic, but also there is back translation of utilizing the emerging clinical data to refine the preclinical hypothesis and guide further preclinical work. So it is a circular process rather than a one-directional process. And essentially what we're doing here is generating plausible and testable hypotheses that can address the perennial development questions in drug development. What's the decision-making biomarker? What's the schedule of the drug? It may not be once daily. If you think about the pharmacokinetics and the pharmacodynamic effects of bisphosphonates, for example, we don't dose according to their pharmacokinetics. Otherwise, we'd be giving it continuous infusions. What disease type? Personalized medicine. How can we reduce the noise to signal ratio to ensure that we see a good enough clinical response? What do we combine with? What are the options to combine with? How do we differentiate from competitors, which I'm sure those of you who work in the biotech industry and also in large pharma will hear from your investors on a regular basis. And then it's particularly in advanced diseases, the disease quite often will progress through the treatment, and what are the next treatment options as far as the patient is concerned. So what's driving this? Well, if we look at an overview of project success rates and the reason for closure, one of the things that is clear is that over the last 30 years we've got better at being able to predict the clinical pharmacokinetics from the preclinical data. So the attrition rate over the last 20 years, as far as PK is concerned, has gone down. But the two key things that show here is that there's still quite a high attrition rate as far as tolerability is concerned in the early development, and still to a degree in late development, but efficacy is also a major driver of drug failure in late development. So anything that we can do to reduce these attrition rates or move everything to the left, either to stop the development of the drug before it moves into the clinic, or to stop development of the drug before it moves into phase 3, will obviously play not only to financial benefits, but also the benefit of the individuals who are volunteering for these studies, and reducing people being exposed to compounds that are not going to become treatments. 2 slides here, just that I quite like with regards to ordering your thought process as you move through from the preclinic into the clinic. Do you have the right target? Do you have the right tissue? Is it the right safety? And obviously, what can be perceived safe in advanced pancreatic cancer is going to be very different to what could be perceived to be safe to treat hypertension. Are they the right patients? Are some patients more likely to see toxicity or more likely to see efficacy? And then how do we differentiate? Even my academic colleagues, you have to have the right commercial potential, because if people don't want to take the drug, or people don't want to reimburse the drug or prescribe the drug, having registration is irrelevant. This is another way of thinking about things again as you move into the clinic. Is the drug getting into the patient? Well, if it isn't, then you may as well pour it directly down the lavatory. But I'm working on a drug at the moment where the 2 tissue compartments to see the least amount of the drug are the blood and the CNS. So tissue penetration and tissue retainment of the drug is also important. Proof of local exposure is really only utilized with the antibiotics. Proof of mechanism, therefore, is a key thing. The drug may be getting into the patient, but is more of it getting into the tissues and more of it getting into the drug. Okay, it's getting into the blood, but is it actually getting into the tumor, which may be fairly hypoxic, and therefore assessing some aspect of either direct target engagement or immediate downstream assessment of inhibition of that target within the organ of interest is important. And then we have proof of concept, and then obviously regulatory endpoints, which in cancer, your proof of concept may be a durable response rate, but the regulators may be looking more for a clear progression-free survival or overall survival advantage. One of the key things that we have to remember is that, as far as translational science, is that killing a drug is not necessarily a bad thing. Attrition during drug development is not a bad thing. What we should be taking forward here are the winners and speeding up the winners. What we should not be doing is delaying cessation of molecules that are not going to make it. And now, obviously, if you have a portfolio of molecules, that is an easier conversation to have. If you only have one molecule within your portfolio, then that becomes a less palatable idea. However, your investors, if you are a biotech with one molecule, that is going to be first and foremost in their mind, which is, if this isn't going to work, I want to stop it as quickly as possible. And it may well be that your drug just isn't doing it what it says on the tin, and either you've got the wrong drug or you've got the wrong hypothesis. It may well be that you have no therapeutic index, or it may be that you have significant toxicity associated with highly variable pharmacokinetics. And unless you could go into a drug monitoring situation, as you would do with gentamicin or warfarin, then there's no real way forward there. It may also be that you have more of a cytotoxic profile, and your drug is supposed to be given chronically. So if you look a little bit more like cisplatin as far as toxicity is concerned, but you want to dose like an aromatase inhibitor, then obviously you need to seriously think about whether that is possible. So just to briefly touch on preclinical pharmacokinetics, we are better at doing it than we used to be, but there is a pressure to move faster into man. People want to get into man as quickly as possible, and I think that is fair. Our discovery medicine PK people are capable of producing much more data than we were with automation and efficient screening, and assuming significant attrition at first into man, because that may well be the only option with the amount of forward movement we've seen in translational aspects of preclinical PK data. Then it may make sense to delay less critical studies like interactions with transporters and what have you. And then there is the question as to how little data can we get away with to get into man. ICHS9, moving into end-stage cancer patients, you need very, very little data. But obviously to move into healthy volunteers or to move into patients with non-malignant disease, you need a larger data package. An idea that I like is strategic implementation of model-informed drug development. So that is to generate a very good preclinical model that can then guide your move into the clinic, and then you can back translate from the clinical data and refine your model and move that forward as you move through the clinic. This potentially can optimize your phase 1 and phase 2 program development. You can develop fit-for-purpose models. And as I said, you can utilize those emerging clinical data to refine this PK/PD model. And although I remain very optimistic with regards to this, unfortunately, I'm not yet to see a good example in oncology, at least, of where that model has actually moved well from the preclinic in the clinic. But I still remain optimistic. Choice of disease type is very important. I'm going to give you 2 examples here of where the choice was incorrect. The first one is SIAC kinase. SIAC kinase is a target that is known to be potentially highly important in cancer. Upregulation of SIAC activity, so you have normal amounts of normal SIAC, but in tumours where it is more highly active, you have a very poor prognosis. In those patients with low SIAC activity, you have a much better prognosis. And SIAC is important in various aspects of normal physiology, cell motility, vascular modulation, cell cycle, etc. And overactivity of SIAC is associated with resistance to a number of targets. The problem we have is that there have been a number of SIAC inhibitors that have moved into the clinic, and really the only one of SIAC inhibitor that has been successful is Dasatinib. But that is more than a SIAC inhibitor. It knocks out c-KIT, PDGF, and various other bits and pieces. And if you look at where it is active, it probably isn't the SIAC that is only driving that activity. And in some cases, SIAC may not be playing a part at all. Now, again, I remain very optimistic with SIAC as a target, but we have to bear in mind that it carries with it the cloud that these compounds haven't been successful. And my own personal view is that it's probably more of an invasion hypothesis, which brings with it the issues of how do you develop an anti-invasive in a paradigm, an oncology drug paradigm that requires tumor shrinkage as a marker of efficacy. Another good example is c-KIT in small cell lung cancer. Again, here you can see from the CT scan and the plots that the c-KIT appears to be a marker of aggressiveness or prognosis as far as small cell lung cancer is concerned. But again, when we look at the studies that have been performed with one drug that inhibits c-KIT, amongst various other things, the results have been very disappointing. And both of these, I think, play to the question you have to ask yourself, which is the target actually in the driving seat, or is that target modulated by other things which are actually driving the disease? It may well be that in small cell lung cancer, c-KIT is modified, its activity is modified, but that is modified by the thing that you should be targeting. In reality, it's probably more complicated than that, which is that it may be c-KIT and a number of things that you have to target to see that activity, because of the inter-exchange between the various pathways that are aberrant within these diseases, and that may well be why dirty drugs tend to work very well in end-stage patients that have very heterogeneous disease. Preclinical efficacy now. So there are a number of models. Here's a focus very much on oncology, and a lot of my examples, unfortunately, are going to be oncology, my area of expertise. And there are a number of very clever models that we can utilize preclinically to allow us to understand the potential efficacy of our drug. But when we look at the shortcomings of those, they all tend to have the same issue. Issues with regards to immune deficiency of these models, and we know that the immune system plays a major part as far as tumor response is concerned. Issues with regards to the stromal microenvironment, and again, we know that that's important in response to tumors, and also heterogeneity. Some of the more modern, more expensive preclinical models will at least play lip service to the heterogeneity that you have within a tumor cell, a particular tumor with certain clones having different drivers, but it doesn't really reflect what you see within the individual patients. And the quote at the bottom here, but somewhat tongue-in-cheek, from Bert Vogelstein, is: the good news is I can cure most cancers as long as you're a mouse, and especially if I've given you the cancer. So pharmacodynamic biomarkers here. So these are proof of mechanism, proof of principle that I'm talking about at the moment. So these are the sort of things that we want the drug to do rather than things that we don't want it to do. And here, the first thing I think we need to know is, does the drug do what it says on the tin? If it inhibits SIAC kinase, is it a SIAC kinase inhibitor? And this can help guide the starting dose as we move into the clinic, and a definition of a minimally biologically active dose, and also guide the schedule. As we said before, if you dose bisphosphonates according to their pharmacokinetics, you would be giving the drug incredibly regularly, but we know that the drug is taken up by the bone, which is the target of organ, or the target organ, and it stays there until the bone is turned over. So you're giving the drug as an intravenous infusion once a quarter or so. So what does it allow us to do? As I've already alluded to, it allows us to delineate the potentially biologically active exposures of the drug. So when do we move from a homeopathic dose to a dose that is more likely to be active? And according to ICH9, we should actually be trying to start our dosing in patients as close to that as possible. And it also allows you to be able to accurately estimate the lower dose range of phase 2 testing. You move into phase 2, and we probably will continue to move into phase 2 in oncology with the dose that is at or round about the maximum tolerated dose, because the mantra of hitting the target as hard as you can for as long as you can is still there. But one of the things that we know, having looked back at the small molecule tyrosine kinase inhibitors, is that quite often we have to drop back from that dose, because what is tolerated for 21 and 28 days in the majority of patients may not be dose tolerated for the long term. Low-grade grumbling adverse events such as nausea, vomiting and diarrhea, okay, they may not stop that short-term dosing, but if you try and live with the nausea and vomiting that you might associate with a hangover every day for 6 months, you're obviously not going to tolerate that. And then knowing that you can drop back as far as a particular point where you no longer see your biological activity, at least guides you with regards to not stepping completely into the unknown. Can also provide the data to support the selection of optimal dosing schedule. And again, I think that the bone turnover markers and the bone mineral density effects of bisphosphonates is a very good metaphor for that. And it can also allow you to be able to make no-go decisions. If you are not hitting the target below the maximum tolerated dose, then again you either have the wrong hypothesis or you have the wrong drug. So a very good example of this is AstraZeneca's pan-AKT inhibitor. And here, during the dose escalation phase and beyond, they looked at 2 compartments. They looked at biomarkers in 2 compartments, and these biomarkers were essentially just downstream of AKT inhibition. So they weren't looking directly at inhibition of AKT directly, they were looking at what happens immediately downstream of that. And they looked at a platelet-enriched plasma, which you could argue is a glorified pharmacokinetic assay, because essentially the platelets are bathed in the blood, which has the drug in. But I still think it's a useful thing to look at. But more importantly, looked in the tumour. And what they saw was a nice dose response, and this helped guide them with regards to the choice of the drug to take forward into the phase 2 part of the experiment, along with the PK and the tolerability data that they had seen. So a very good example of where they knew where their minimally biologically active dose was, their maximum tolerated dose, and therefore they were able to move forward with a high dose, but also an understanding that if that wasn't tolerated, they could drop back down to something that was still biologically active. So the 3 biomarkers approaches here are proof of mechanism, does it hit the target in man? Proof of principle, does it have an effect on the disease phenotype? Just hitting the target isn't enough. If it's an anti-proliferative, you should see a reduction in something like Ki-67. If it's a pro-apoptotic, you should see a change in apoptotic markers. And then does it have a clinical benefit as far as the physician at least is concerned, such as a change in tumour size reduction, an overall reduction in tumour growth, resulting in a clinical benefit rate. Does it give you guarantee of success? No, it just allows you to move forward with increasing levels of confidence with regards to your outcome, and it plays back to the tool in the Smethurst and Hughes paper, which I strongly recommend that you read. Toxicity. Well, again, if we can kill drugs before they move into the clinic because they're going to be toxic, we should be doing so, but we should also define markers of later toxicity or more acute toxicity that can allow us to be able to pick that up early. And I'm going to give you an example of some pitfalls here. So here is a Johnson & Johnson's oral MET inhibitor that moved into the clinic a while ago. They'd done their preclinical tox in the rat and the dog, and not seen any renal toxicity at all. As you can see from the top plots here, they moved into the clinic and they saw rises in serum creatinine and reductions in glomerular filtration rate. So this drug was having a negative effect on renal function. They looked closely into that and they found that what was happening was that a metabolite driven by aldehyde oxidase activity was precipitating in the kidneys and causing renal damage. Aldehyde oxidase isn't particularly relevant in the rat and the dog, but it is highly relevant in the kidney and highly relevant in man. So essentially they chose the wrong tox species in retrospect. It's always easy to say these things in retrospect. So choose your model very carefully. Here's another drug. This is a SIAC kinase inhibitor. Renal function in healthy subjects as measured by creatinine. And again, as you can see here, a huge increase in creatinine levels in these healthy individuals. So should we be killing this drug? Is it causing nephrotoxicity? Well, Saracatinib is an OCT2 inhibitor, and OCT2 is required for secretion of creatinine at the distal tubule. If you inhibit OCT2, as large numbers of compounds do to a certain extent, including ranitidine and cimetidine, you will see increases in creatinine with absolutely no effect on glomerular filtration rate. So should we have stopped the development of this drug because of renal toxicity? No, because it's not nephrotoxic. It's an OCT2 inhibitor. We were just looking at the wrong biomarker. And in the presence of an OCT2 inhibitor, well, frankly, I don't think highly of creatinine as a marker of renal function anyway, personally. But in the presence of an OCT2 inhibitor, it is not much use at all. And the FDA have published on this, and one should be looking at markers such as cystatin C, which is has a similar origin as creatinine, but is only filtered at the glomerulus, and then KIM1, which is a marker of renal, a direct marker of renal toxicity. The problem you have is that those aren't routinely assessed in the majority of hospitals. Predictive biomarkers. Now, this is really where translational medicine has speeded up drug development rather than just acting as a killer of drugs that aren't going to make it. And here again, we've got AstraZeneca's pan-AKT inhibitor, and if you look at the response data on the left of this slide, you don't get particularly excited. If you then look purely at gynae tumours, so breast and pelvic gynae tumours, you're getting a little bit more excited, but there still doesn't seem to be a good correlation. However, if you look at PIK-C3A mutation, you now actually get quite excited. You now have a selection tool. An enrichment tool, clinical enrichment tool, would be gynae tumours and breast tumours, but your selection tool is PIK3CA. That is where you're seeing your responses. Okay. Another good example is osimertinib. This molecule was specifically designed to target T790 mutations in EGFR, which were the major driver to the first generation EGFR agents. And from the outset, AstraZeneca looked at T790 mutations in the patients, and therefore they saw a very high durable response rate in their phase 1 studies, and essentially had accelerated approval in that subpopulation based on 2 beefed up phase 1 studies. There are a number of other drugs that have done this. The immuno checkpoint inhibitors also had accelerated approval. The problem there is that PD-1, PD-L1 expression are probably only enrichment tools for response to the checkpoint inhibitors. The actual clinical benefit is broader, and therefore the survival advantage is broader than that. And that is why in diseases like head and neck cancer, and also more recently the indications have been broadened within lung cancer, and the cutoff has been dropped for expression. It is because we really don't know exactly why some patients have a clinical benefit to these agents in the absence of any real PD-1, PD-L1 expression. So combination strategy is another one. Some drugs obviously will move forward with a monotherapy hypothesis, but there's the opportunity for additive and synergistic effects in combination. Here's an example again of why we shouldn't have done a study. This is the ATAC study that I was involved in a long time ago when I first moved into industry. Here we had 3 arms in the adjuvant treatment of women with ER-positive breast cancer: an anastrozole itself, which is an aromatase inhibitor, tamoxifen, which is an estrogen blocker, and a combination of the two. And as you can see from the little table on the left, we recruited over 3,000 patients into each of these, and then the results showed that actually the combination was no better than tamoxifen itself. Essentially, at the very low estrogen levels that you see in a postmenopausal woman with an aromatase inhibitor, tamoxifen starts to act like a partial agonist, so it has somewhat of a stimulatory effect. So you therefore lose that synergistic of the additive, the advantage rather that an anastrozole has over tamoxifen. So that was a lot of patients to put through that study for essentially a wasted arm in this 3-arm study. Here's some data that we had at the time we started that study, which shows clinically that the effect on Ki-67 with the combination isn't really any better than tamoxifen. So we actually had the data not to perform that arm at the time we started that study. The reasons for doing that study were the reason for that extra arm in the study were the reasons that that many pharmaceutical companies have fallen into this particular trap before. People get excited by data and hypothesize data and miss data that may be in the database. But the translational science data said that that combination arm was not going to be successful, and it wasn't. Differentiation strategy, this is key. I think to all of us that work in industry, we're always being asked why our molecule differs from not only the combination, the comparator drugs that are out there in the clinic or being developed, but also from the other drugs within a portfolio, if you are lucky enough to have a portfolio of drugs. Here, one of the questions again with fulvestrant was, well, is it actually any different to tamoxifen? You've got the control sample there from a patient that a breast cancer that hasn't been treated with anything as yet. At the bottom, you can see, and you can in that top side, you can see the cells are stained for the estrogen receptor. And you look at the bottom left where the patient has been treated with tamoxifen, the estrogen receptor is still there. With fulvestrant, however, that staining is gone, and that is because fulvestrant not only blocks the estrogen receptor, which is what tamoxifen does, but it also causes degradation of that estrogen. It's a down regulator of estrogen, so therefore it does have a different effect. And there you go again. There's data from the clinic that shows that as well. Another more recent example of PARP inhibitors. Now, up until recently, we thought that one PARP inhibitor was very much the same as another, but since then we've realized that not only do you have PARP enzyme inhibition, but you also have PARP trapping, which also you would expect to be associated with an at least additive, if not synergistic, activity to the enzyme inhibition. So when you look at the preclinical data, and here's talazoparib, which is the BMN637 molecule, compared to olaparib and rucaparib. And as you can see from this, it does appear to be the most potent inhibitor with the highest efficiency of trapping PARP DNA. But what does that mean as far as preclinical cytotoxicity is concerned? Well, again, as you can see from the plots on the right, it does appear to be markedly more cytotoxic than olaparib and rucaparib. Now we know from the clinic, because this molecule isn't quite there yet, that olaparib and rucaparib already differ with regards to their licensed indication. Olaparib requires patients to have a BRCA mutation, whereas rucaparib has much broader a broader indication, probably playing to the fact that a lot of these patients, although not having BRCA itself, have other DNA damage pathway aberrations that result in something that is referred to as BRCA-ness. But what we do know with regards to this effect is that as far as the clinic is concerned, it does appear that this particular molecule is associated with more bone marrow toxicity. So what you may well be doing with this PARP trapping is turning cells that do not have a BRCA-ness phenotype into having a BRCA-ness phenotype. And that isn't only the tumor cells, but may also be other cells within the body that are rapidly dividing, like the bone marrow. So this extra efficacy may bring with it extra toxicity. Acquired resistance. All the treatments that we give, particularly in advanced cancer, will fail. And what we need to know is, what do you give next? So here, again, playing back to some more historical data, one of the things that we found was that preclinically we would have expected fulvestrant to work in patients that have failed on tamoxifen, and that was recapitulated in the clinic, where we did see a significant activity of fulvestrant in patients that have failed on tamoxifen. But something a little bit more sexy and more recent is circulating free DNA testing. So this is where you're looking at DNA which is shed from a tumor in the patients. And here you can look at the evolution of the tumor burden, the genomic changes within the tumor burden over time. So this top one here is a patient that's been treated with everolimus, and as you can see, the blue changes, the PIK3CA burden in this patient goes down, but you see an upregulation of ESR and other drivers, genomic drivers. Okay, so you're beginning to see as the disease evolves through that therapy. And again, here at the bottom, you've got a molecular evidence of disease at the outset, and people are beginning to talk about potentially using circulating free DNA as a diagnostic for malignancies. And you see that heterogeneity evolve, and then you can measure the molecular response to that drug, and at the end there you can see a certain clone growing through and becoming the majority of the circulating tumor burden. And to go back to osimertinib again, this has been used with osimertinib and the other EGFR inhibitors, and here you can see the effect of a downregulation of EGFR mutations as the patient receives the osimertinib. But you can be with this patient, you can see particularly PI3 kinase coming through as a potential driver of that resistance. And this sort of work has resulted in an understanding that MET amplification plays a large part in resistance to the EGFR TKIs, particularly osimertinib, and has resulted in a number of companies with MET inhibitors combining those MET inhibitors with EGFR patients in an attempt to cause responses in patients that have failed the EGFR inhibitor because of MET aberrations. So what's the take-home message from this? Well, we have large number of tools that we can use preclinically to generate hypotheses preclinically and take those hypotheses into the clinic, where we can test them and then take the results of the clinical data and back translate those into the preclinic. And this can be genomic, transcriptomics, proteomics, cytometrics, metabolomics, and these allow us to get a multi-dimensional understanding of the disease pathology, and again in oncology particularly an understanding of the heterogeneity of the disease. We say that the patient has ER positive breast cancer. That means that the patient has a large number of clones within their tumor burden that is ER positive. They will also have other clones that have different drivers to lesser or greater percentages of that clonal burden. And as you treat one set, the others will grow through. So we get an understanding of that. The issue you have is that you do have to choose your tool carefully, because garbage in is garbage out. So therefore, it has to be you have to have a robust biomarker discovery, development, and validation effort that can then allow you to be able to choose the correct tool to answer the question that you're answering. And it may be, unfortunately, that tool does not currently sit on the shelf. It may be that it doesn't currently exist, and that's often the case with novel targeted agents as far as a proof of mechanism, a target engagement, direct target engagement measure. Does the drug do what it says on the tin? And in that situation, you may have to develop and validate your biomarker yourself. Or there may be an on-the-shelf biomarker that allows you to be able to assess an immediate downstream effect of that in that target inhibition, as the AstraZeneca's AKT inhibitor utilized. That doesn't mean you have to validate it, because validating a biomarker is a very long and complex and expensive thing to do. So if you do not have a biomarker that is on the shelf, then you're going to have to go to your investors, your payers, the people who are funding your study, and request that funding. And the time to generate that is quite long. So the best time to do that is probably around about lead optimization. And I can tell you, you will get pushback on that because, especially in a small organization, that funding usually hasn't been built into the process. And even in large companies, they would prefer to be able to take something off the shelf. The next point with regards to translational science is, as I've already alluded to, the key place that we can play to our strengths is to pick the winners early and to kill the losers early. And unfortunately, there are usually more losers than there are winners. So translational science can sometimes be very negatively viewed because all we do is kill molecules. Yeah, but these are molecules that we're going to die later. We've killed them earlier. And that is quite a difficult thing for the people who are measuring success, because what they're measuring is negative success. So we do have a problem within translational science of how we are measured and how we are perceived. Now that is something that we will work on, and I think that, you know, when you talk to the funders, and you talk to people at strategic level within large pharma, they get this and they understand that what we're trying to do is to utilize the resource that we have the most efficiently and focus it on those molecules that are going to be successful. As we do this more often, and drugs are accelerated through to indications, particularly piggybacking on the accelerated approval strategies that the FDA and to a lesser degree the MEA have, I think that we will be perceived more positively. But I've been doing this now for 15 years, and sometimes you're in the situation of perceived to be somewhat of a harbinger of doom, and wearing a black cowl and scythe. So that's pretty much in a nutshell. There are lots of things that we can do. There are lots of tools that are out there, but you have to understand the question that you're asking. You have to choose the right tool to do that, and people also have to understand the impact of the answer that you may give, because it may not be what they want. It may well be your drug isn't doing what it is, it's too toxic, we have the wrong schedule, the dose is too high in phase 2, we need to drop down. And these are all things that might be perceived as derailers, but they're not derailers, because they are going to either stop the development of a drug that shouldn't be going any further, or answer questions that I would rather answer in phase 1 or early in phase 2 than be tripped over in phase 3. Okay, any questions?
Hi there, it's Georgina from the BIA. I just want to say thank you very much, Glenn. That was an excellent presentation. And just to the audience, if you have any questions, there's a question box in the control panel in the webinar program, and you can just submit them there. And Glenn, we will read them out for you to answer. So there's one here to get started with, and I would encourage anyone, any question at all, please do submit it for Glenn. So the first is, when should the translational science or clinical team get involved?
So this is a key point, because the response to that question is usually you've missed the boat. It should have been 2 years ago. But as I've already alluded to, I think the key point at which you need to engage your translational science team, and that should be a mixture of your preclinical experts and your clinical experts, is at the time of lead optimization. Because if you need to have some of these biomarkers, you need to start understanding what they are and whether they exist at that point. And if they don't, you either need to work them up yourself, or you need to collaborate with an academic group, or perhaps a clinical research organization that have an interest in this area, to work up those biomarkers and validate them and get them clinic ready and physician proof. And that will take a couple of years at the very least. So lead optimization is that point. And quite often, unfortunately, people engage the translational science and particularly the clinical aspect of translational science, when they've delivered their GLP tox data, and that is far too late in my view. And then you really are scrabbling around to try and pull together a package to be able to support your first into man study.
And we have another question here as well. Shouldn't we be hitting the target as hard as we can for as long as we can?
Well, again, I mean, a good example of this is the bisphosphonates. You could do, but then everything is a poison. It's just dependent upon the dose. And if you only have to downregulate the target for 8 hours of the day, then anything over and above that is going to result in toxicity. A lot of these pathways are important in all cells within the body, not just the tumor cells or the aberrant osteoclast, osteoblast activity that you may have in in arthritis or osteopenia, that's true for a cytotoxic drug. For a platinum-based chemotherapy agent, we know that you have to sail as close to the wind as you can with regards to the toxicity to maximize the efficacy of the drug. But if you look at 5-FU, there is a huge therapeutic index with 5-FU. We could give patients much more of the drugs, and sometimes do, but that is very rarely associated with an increase in the efficacy of that drug, and is associated with an increase in toxicity. And what we have to remember about more chronic diseases, particularly end-stage cancer, is that it's a balance of trying to extend the patient's life by minimizing the impact on quality of life. These patients are not going to be cured by our treatment. This isn't like early breast cancer, where you may well be curing your patient by giving them their radiotherapy and their chemotherapy post surgery. These are patients that are either going to die of their disease, or probably less likely die of something else some sort of comorbidity before they die of their disease. The problem that you have is that there is that mentality. There is still very much a cytotoxic mentality amongst clinicians, amongst oncologists, and particularly amongst the funders that we have. So will we continue to move into the clinic with something that is close or at MTD? I think we probably, unfortunately, will do, because people will look at the biomarker data and say, that's very interesting. But by having an understanding of the biological activity as you go up through the doses, when you do find some toxicity issues hopefully in phase 2 and not in phase 3, you know what you can drop back to and still have an active dose. That doesn't mean to say it's going to be as clinically active perhaps as it was, but it does give you that ability to do so. My hope is that over the next 10 years, we will be much more clever in how we choose our dose, and we will utilize the biomarker data as we better understand it, and we better understand its relationship to clinical activity. But at the moment, in oncology at least, it is primarily the toxic tolerability of the drug, the short-term tolerability of the drug that drives the choice of recommended phase 2 dose. Okay.
Brilliant. So I think we're limited on time, so we'll just finish up with this final question here, which is, do we really need a patient selection biomarker?
Yes, so I often get this one. If your drug is likely to work in 60–70% of patients, then no. I think that the IO agents were lucky. Their response rates were 10–20% at most as far as monotherapy is concerned. And let's face it, docetaxel's activity in third-line lung cancer is around about 20%, okay, which isn't particularly good. So I think they were lucky. But I think people these days are expecting higher and more durable response rates with the treatments that we're giving. Okay, we can't go with what we've done in the past, and to do that, I think you do have to at least stack the cards in your favor and have a patient enrichment tool or a patient selection tool. If you go with a patient selection tool, the drug may work more broadly, but if it doesn't work in the acid test population, let's say, if osimertinib hadn't worked in the T790 population, it wouldn't have been taken forward. It may well work more broadly than that, and it does work more broadly than that. But if it hadn't worked there, it would have failed the acid test and it wouldn't have moved forward. So if you do have a patient selection tool, which is your primary hypothesis, then at the very least enrich for that patient population so you can see that it does what you expect it to. If it works more broadly, then that's great. If it doesn't work in your patient selected population and it works in the other patient population, that's fine, but your hypothesis is wrong. So I think you need to think about a way of enriching your patients. Otherwise, you will lose any signal. You have the potential to lose any signal that you have within the noise of the broader population that you've chosen. So that's always my starting position. Okay.
Fantastic. Thank you so much, Glenn. And just a couple of words from us to finish up. We are the BIA, the Trade Association for Innovative Life Sciences in the UK. And if you are listening and you're not a member, but you're interested in joining and what we do, please get in contact or visit our website for more information. This has been episode 3 in our Bench to Bedside webinar series. And we hope you will all join us again for the final episode on the 11th of October and for our concluding panel session at the UK Bioscience Forum on the 17th of October. So for more information and to register for the next episodes or the Bioscience Forum, you can visit our website and find our events page at www.bioindustry.org. Glenn, thank you so much for joining us today. I think it's been a really wonderful presentation and very thorough. Thank you also to everybody in the audience who's joined us, and we look forward to reconnecting for the next episode on the 11th of October.
Great. Thanks a lot, guys, for your time. Take care. Bye.